Welcome dear students. I am here to deliver the lecture on the topic. The subject is Human Anatomy and Physiology-II. Subject code is BP201T. And today's topic is chromosome, gene and DNA. In the previous lectures, we discussed about the entire system of the reproductive system. And here we discuss about the structure of the chromosomes, what is the function of chromosomes, what is genes and what is DNA and what is the structure and the functions of the DNA. Now start with the introduction. So what is genetics? So basically genetics, all the cells have a capability to give rise a new cells and they encoded the information in a living cells is passed from one generation to another generation. This information encoding material is the genetic or hereditary materials of the cells. Means genetics is the information which is transferred from the one generation to the another generation and it is the hereditary character of the person. Okay. Then what is genes? So genes is a unit of inheritance that are located on the chromosomes on the gametes, which is passed from the parents to the progeny, or it's a pass from the one generation to the another generation. So this is the gene. So gene is the unit inheritance. Now chromosomes. What is chromosomes? Basically we always heard about the chromosome number. So basically chromosome is a thread-like structure of the nucleic acid and proteins found into the nucleus of the most of the living cells carrying the genetic information of the form genes. So what is chromosomes? Chromosomes is a thread-like structure and which carry all the genetic informations of that particular gene, and the number of chromosomes is fixed but it varies from species to species. The number of chromosomes is always fixed, that in the human it is 23 pairs or there is a 46 number of chromosomes in the human. So in human beings their total number of chromosomes is 46 or it is present in the pair, so it is a 23 pair, it known as the diploid, right. So this chromosomes is a 2n, that is diploid in nature. Now while in a gametes, male or female, there is the number is 23, means if a 23 it is a haploid, if a 46 is a diploid, it is a n, 23 is equals to n and 46 means 2n. It is a haploid and this is a diploid, right. So there are 23 pairs, right, but here there is a one important point that you have to learn, that is a 20 pairs of chromosomes are known as the autosomes. Okay, there is a 21 pairs are known as the autosomes and there is a single pair that is known as the sex chromosome. Means 22 pairs is known as the autosomes and the one pair it is known as the sex chromosome, and the length of that chromosomes are 4 to 6 microns. Then the structure of the chromosomes. So there are the following components present inside the chromosomes. So there is a first centromere. Centromere means it is also known as the kinetochore and it is a primary constricted at the center where the chromatid and the spindle fibers are attached. Okay. So these centromeres are present on the center and these centromeres are also known as the kinetochore. Its function is in the movement of the chromosome during the stage of anaphase during the cell division. So the function of the centromere is for the movement of these chromosomes during the phase of anaphase division. Now, next one is the chromatid. So, chromatids, when the chromosomes are divided into the two identical strands during the cell division, the chromatid is formed a half of chromosomes, means half these chromosomes in the pair, right, that is a 2n, but when they form they are single in number, means they are haploid, they are just half. So, they are haploid. So, chromatids form as a half of the chromosome. Each half strand is joined by the centromere, both known as sister chromatids, and it contains DNA and separate at anaphase to separate from chromosome. Now, next one is the chromatin. So, these chromatin is a complex DNA consisting of the DNA, RNA and proteins, and it forms the chromosome within the nucleus of the eukaryote cells, and it does not exist freely or it does not exist freely as linear, rather it is highly condensed and wrapped around the nuclear protein, means these chromosomes are not really free to move because they are coiled nearby the nucleus. Now, last one is a telomere. So, telomere, the two strands of the chromosomes are called as the two ends. The two ends of the chromosomes are known as a telomere. These telomere are highly stable and they do not fuse and unite with telomere to other chromosomes. Okay. So, here there is a structure of the chromosomes. So, always remember that these two half is short and these lower structure is long in shape. Okay. So, there is a structure of the one chromosome. So, these two wings, these are the two identical chromatids which contain an exact copy of one and each other contain one DNA molecule, and this is called as a P arm. Okay, this is called as a P arm, it is short in its structure, and this is a centromere. This is a centromere which is constrict the point, which is a constricted point of the centromere. It is the center part of the chromosome, and this lower part, it is known as the Q arm. So, Q arm is longer than the P arm. So, this upper arm, this arm is known as the P arm, and this lower arm is known as the Q arm, and these are longer as compared with the P arm, and here it is present, that is a DNA. So, DNA molecule, long strike like DNA molecule formed into the compact structure by the protein called as histones. So, they are the histone proteins which are formed further the DNA molecules. So, this is the structure of the chromosomes, right. Now, there is some positions, there is some classifications of chromosomes. So, there is a classification of chromosome according to their position, or it is according to the position of the centromere, means if the centromere is present in the center or it is present on the entire surface of the chromosome. So, it is divided on that basis. So, first is the telocentric. Okay, first is the telocentric. So, here the centromere at the tail end, that is it's a centromere, it's a chromosome arm, that is a acrocentric. This is acrocentric, where the chromosome shift towards the p-arm, means during the this acrocentric, the centromere shift towards the p-arm, it means it is shift towards the upside, right. This is about satellite chromosomes. So, and next one is submetacentric. Submetacentric means it is nearly to the center, nearby the center, and last one is the metacentric, where it is just exact center part of the chromosome. Okay? So, there is a telomere, when the centromere is present on the end part, then acrocentric means when the chromosome push or it is present towards the p-arm or it's towards the upside of the chromosome structure. Then submetacentric it is up to the it is again upside, and next one is the metacentric, it is just center part of the chromosome. So, these are about the classification of the chromosomes. Now, there are the various types of chromosomes, right. So, there are the various types of chromosome, that is a homologous chromosome. Homologous means when the two substance is identical to each other, then two are same, that is a homologous. Okay. So, homologous chromosome pertain the two chromosome which are same at least in the term of the gene sequencing or the loci. And what is heterogeneous? Heterogeneous means differ. So, heterogeneous, it's heterogeneous pertain to any two chromosomes that are different, such as in the term of the gene sequence or the loci. So, here there is a homologous pair, means chromosome forms the mother and this chromosomes from the father, and both chromosomes are identical to each other, they are same to each other in the sequencing, in the condition of the gene sequencing or in the condition of the loci. So, it is the structure of the homologous gene. Now, chromosome numbers. So, as we discuss, as I discuss later, that is there is a different numbers, as if single, it's a haploid, it's a double, it's a diploid, and there is a number of chromosomes, means there is a 46 number of chromosomes which are present in the pair, that is a 23 pairs of chromosome. So, it is of two types, that is somatic chromosomes number, that is a 2n, any chromosome that is not a sex chromosome appear in the pair in a body, but as a single chromosome in spermatozoa. So, what is somatic chromosomes? Any chromosome that not a sex chromosome and which appears in the pair of the body, but as a single chromosome in the spermatozoa. Next is a gametic chromosome number, that is N, it is a haploid. So, in human gametes, our haploid cells contain the 23 chromosomes, each of which have one chromosome pair that exists in the diploid cell. The number of chromosome in the single set is represented as the N, which is called as a haploid number. In human, it's an N is equals to 23, it's a haploid. If it is in a pair, so it is 23 pair, means 23 plus 23 is equals to 46. Okay. Now, functions of chromosomes. So, there are the various functions of chromosomes. So, chromosomes basically carry the basic genetic material or genetic information in DNA, which is responsible to provide the hereditary character of the one generation to the another generation. And this characteristics and genetic information is various to the various body or the various cells. The cellular functions are important for the growth and survival of the living organism. And the chemical composition, if we talk about the chemical composition of the chromosomes, so they are covered with the histone proteins and other proteins whose role is to protection from the chemical enzymatic activities and the physical forces due to which the chromosomes may be damaged. Okay. So, in this, in turn, it helps the DNA to have a safeguard during the process of the cell division. So, these are the histone proteins or some other proteins which are work as a DNA safeguard to in the process of the cell division. Then there is some non-histone proteins present in the chromosomes which helps in the regulation of the gene actions. Now, this is all about the chromosome. Now, next one is a DNA. So, the full form of the DNA is a deoxyribonucleic acid, that is a DNA. So, DNA is a herited material found in the every single living thing, and this DNA is individuals chromosome control a variety of visible characteristics and invisible characteristics. Okay, so sometimes the condition or the hereditary material or the genes are dominating in nature, dominates, and sometimes it is recessive, but that means recessive means that doesn't show at that condition. Okay. So, sometimes the conditions of the variety is shown, that is the dominance, or sometimes it is not shown, it is hidden, so it is recessive. Okay. So, visible include race, coloring and sex, and individual characteristics including blood group and susceptibility to the inherited diseases. Now, DNA is made up of the molecules called as nucleotides. Each nucleotides contain a sugar group, a phosphate group and the nitrogen bases. So, basically the DNA, it is made up of the nucleotides contain a sugar group, phosphate group and the nitrogenous bases. With all these components, the structure of DNA will form. Then the deoxyribonucleic acid is same in all the cells in the body of individual, but there are some sequences, there is alteration in the some sequences. So, DNA has a double helical structure. Double helical means it is just like this. This is single helical and this is the ladder shape, means there is a ladder and this is just rotate that ladder. Okay. So, it is a double helical structure. The outer edges are formed the altering deoxyribose sugar molecules and phosphate group, which is make up of sugar phosphate backbone. So, this sugar phosphate this is made up of the sugar and phosphate bonds. Okay. The two strands run opposite directions. This means the nucleotide in each strand DNA are exactly complementary to one than on the other strand. Now, next one, the four types of nitrogen bases. So, there are the four types of nitrogen bases. There are adenine, thymine, guanine and cytosine. Okay, adenine, thymine, guanosine and cytosine. But if we talk about the RNA, so there is a cytosine replaced with the uracil. Okay, so this replaced with the uracil. So, that is the structure of the RNA, but here we discuss about the only DNA. So, DNA mein the first is the adenine. So, adenine and thymine are the pair of two hydrogen bonds, where the cytosine and guanine are paired of the 3 hydrogen bonds. So, this adenine and thymine is the 2 hydrogen bonds, and this guanine and cytosine have a 3 H bonds, yeah, the 3 hydrogen bonds, and the DNA is same in all the cell bodies of the individual. The DNA is a double helical structure. The outer edge is made up of the sugar and phosphates. So, nitrogen bases are positioned inside the helix, means this is, if this is the helical structure, so the nitrogen bases present inside this A, T, G, okay, C, G. So, means if we talk about the structure of DNA, So all the nitrogen bases are located inside these double helical structures, and the bases located inside the double helix are stacked. They are twisted or turns into the 2 sugar phosphate backbone of the double helix occurs every 10 base pairs, which maximize efficiency of the base pair packing. So this is the structure of the DNA. So if this is the base pairs, means this is a base pairs, means this is a nitrogenous base pairs, okay. And this outer layer is made up of sugar and phosphate backbone. And if you are then we talk about in the structure, means we open this structure. So there is some nucleopeptides, and this is the nitrogen. This is a nitrogen basis, and it is connected with the hydrogen bonds, okay. So the adenine and this thymine have 2 hydrogen bonds, but alternatively the guanine and the thymine is the 3 hydrogen bonds. Now functions, if we talk about the functions of the replication, function of the DNA. So the function of the DNA is, first is replication. Replication means to form the multiples, replicate, okay. So DNA exists in a double helical arrangement in which each base along one strand binds to the complementary base on the other strand. That is, T's can only bind with adenine, means thymine always bind with the adenine, and this cytosine always bind with the guanine, okay. So this is the specifications, means A always bind with T and and C always bind with the G. And this and in the bacteria and the mitochondria may also have a single stranded DNA arranged in a ring. When a cell divides, the chromosomes containing the DNA strand must be replicated, or it makes the copies of themselves so that the both daughter cells receive the full genetic material, okay. So during the replication condition, the DNA double helix unwind along each strand to the template for the newly synthesized complementary strands to form the new double helix. The enzyme DNA polymerase assists in the process. Now next one is the encoding information. So encoding information, it is again, encoding means ATGC. So these are arranged, and the sequence in which these are arranged, the sequencing according to these arranged, it shows the genetic characteristics of that particularly living being. Now mutation and recombination. So this DNA also in the mutation and recombination means this chromosome DNA helical does not usually interact with each other. However, through the process of the genetic recombination, segments of differentiation chromosomes swap the place with the creatives, okay. Then if there is some changes occurs in the DNA sequencing of the sex cells, the changes can be inherited by the next generation. If the changes will done on the current generation, then they continue with the next and next on generations, okay. The new sequencing might produce a new protein, some of which are beneficial to the organism. Then in the way of characteristics of organism, that is the might involve the time. The mutation help with the illegal base pairing. Mutation means the sudden changes. So there is a specific segmentation, as I told you, that A always compared with the T and G always paired with the C. But there is a mutation, means there is an illegal pairment, means it may be combination of the wrong pair, means A combined with the U or A combined with the G, Okay, so it will mutate the offsprings. Then now next is the gene expression. Gene expression means each cell contains the full complement of a gene, yet the cell forms the different tissues, organs look like, behave differently. DNA plays a traffic cop of the types of the proteins which does not interact the protein cells. Okay, the DNA responds to neither particular proteins, exposing the gene inactive. Okay, so these all are the functions of the DNA, the structure of DNA and about the genes. Thank you.